we will have the next speaker, Julie, to introduce a new methods as a new product we just launched. So Julie is the director of R&D at Singleron, and she has a long experience developing NGS and single-cell products. So she worked at Takara and Swift Science before joining us. And today she will present one of the methods to multiplex your samples without bias based on the click chemistry. So yeah, welcome, Julie. Stage is yours.
Thank you, Yuan. Today is my great pleasure to introduce you to the newest product by Singleron. It was launched only a couple of weeks ago. The product, as Yuan just described, is used to multiplex different samples for your single-cell RNA-seq experiment. So let's dive in. So before I go into why you should multiplex your sample for RNA-seq experiment, I want to introduce a little bit our JAK-scope workflow. So basically, it starts with your tissue of interest or your biopsy or whatever cells that you want to use. Singleron came up with a tissue preservation buffer that will allow you to keep the sample alive for up to 72 hours. So that allows you to collect different samples and you can then multiplex them all together. We also offer a good tissue dissociation buffer that allows the dissociation of all the cell in single cell, of course. And what is particular about this dissociation buffer is that all the cells are going to be efficiently dissociated from each other. So that means that if you have a cell type that is more tightly attached together, well, you won't lose those cells because we are able to make them available for single-cell RNA sequencing. In the middle of the workflow, and I think it's one of the best feature by Singleron, is the matrix. This machine is all automated, and it allows you to perform the partitioning of your cell. It also does the cell lysis, also the barcoding, and the capture of your messenger RNA. So it's very cool, and it only lasts 40 minutes, so it's super fast. Singleron also offer reagents to make your cDNA and your library construction for Illumina sequencing. And finally, we also offer analysis, bioinformatic analysis software named CellScope that will allow you to have a better picture of your data on the visualization aspect of things, but also having a database to query of. Okay, so coming back to our topic of today. So why would you like to multiplex a sample to go into a single cell experiment? Well, obviously, it's to simplify the workflow. So imagine that you have 6 different samples that you collected in a week, and you want to process all of them. So with multiplexing, you only have to run the whole workflow once. It reduces the cost, of course. If you want to multiplex 6 samples at a time, you only have to load 1 chip to do 1 cDNA and to make 1 library. So you save time, you save money. It also reduces batch effect. So it's easy to understand that if you have the sample all processed together the same day by the same operator with the same reagent on the same instruments, that the variation are going to be reduced greatly. The whole process also enables better identification of doublet and multiplet. So you will know if 2 cells fell in the same well in the microchip because they're most likely going to have 2 different tags. So you can remove them from your analysis. And one of the other things that I was thinking that people could do with the kit is to introduce, for example, a spike-in of a QC sample. So if you're new to single-cell sequencing experiment, you want to make sure that your process is great and consistent between experiment, you can introduce a small spike-in of some cells that you're sure of, that you know that they behave well. And also you can include some more challenging sample. So for example, again, if you have 6 samples and one of the 6 samples doesn't look as good that day, but you would still want to introduce and have some data, well, you could still include that. Of course, it's some risk to it, but you may have some data out of it. And the 5 other samples that are performing well will enable you to have a good cDNA and a good library. Okay, so how does that work? So the first step is, of course, the sample tagging. So if you have, for example, here 3 different samples, all the cells inside the same sample will receive the same tag. Here they are represented by the different color, orange, blue, and purple. You can have up to 16 different tags in the same experiment. The tagging is led by the click chemistry. I'm going to talk about the click chemistry a little bit more in detail in the next slide. But let's see a little bit more about the Gexscope workflow. So once your cells are tagged, then you pull them all together, and you can choose the different ratio depending on what cell you want to see, or you can just basically load all of them at the same ratio. You then load the cell on our microchip. So that's the figure in the middle of your screen. You inject, or you use the matrix for automation, but you can inject your cell in one of the little pore here on the flow cell. Then you will add the magnetic barcoded beads that will enable the cell to be barcoded. Then the cells are going to be lysed. And doing so, the messenger RNA, as well as the CLindex sample oligo tag, are going to be released. And if you go to the figure on the right, you will see that they both contain a poly A sequence. And that poly A sequence will bind the poly T sequence that is on the oligo that is coupled to the magnetic beads. The oligo on the magnetic beads also contain the cell barcode. So both the CLindex sample tag oligo and the messenger RNA receive the same cell barcode. And during the reverse transcription, the poly T tail there is going to be extended, and you will have 2 different duplex that are going to be formed. So the first one will be a messenger RNA DNA duplex, and the second one will be a CLindex sample tag oligo DNA duplex. Then we go into amplification of the 2 products by a sample PCR. So we have a lot of those molecules that are amplified, and we separate them by size selection. So the CLindex tag oligo is about 200 base pair, and the cDNA is about 1500 base pair. So it's a great difference between the 2. And using samples price select or impure beads, you can basically separate the 2 products. So the supernatant will contain your CLindex tag, and the beads will retain the cDNA. To make an Illumina compatible library prep with the CLindex tag, it's really easy. We basically take the product there, and by a sample PCR, we incorporate the Illumina adapters. For the cDNA, we first fragment the cDNA with an enzyme, pardon me, and then we ligate the Illumina adapter, and we amplified and enrich for the 3' end of the messenger. You can see the size distribution of the 2 products is really different. So on top, you have a sharp peak, which is the CLindex product, because it's always the same size. It's only 15 or 16 base pair, and it's always the same. For the cDNA that is made into the library prep, it's a nice bell curve distribution. You then pull together the 2 types of library at a different ratio, because you want to sequence way more of your messenger RNA that you want to sequence the CLindex. So for a standard chip, for example, we will use a ratio 1:20. For a high density chip, I can imagine that would be something more like 1:40. And you then proceed to analyzing your data bioinformatically. So the cells in your experiments are going to be grouped by sample using the CLindex tags, but the messenger are going to be grouped together by cell using the cell barcode. And because the cell barcode is found in both the CLindex tag library as well as the cDNA library, you can then, as a common denominator, put them back together. Hopefully it makes sense. Okay, let's talk a little bit more about the click in CLindex. So it's a really simple process. So basically, you only need 3 different things. You need wash cell, about 1 or 2 million of them. You need a chemical group that we provide to you. And this chemical group likes protein at the surface of the cell, any protein. And they also have 16 different tags that are also going to be bind to this chemical group. So in a click, adding a quencher, the chemical group will bind the cell, and the ClinTag oligo will bind the chemical group. They are covalently attached, so it's a permanent binding there. So you can wash the cell, you can go into fax. Nothing is going to basically detach that tag from the cell. It takes about 30 minutes upfront, the conventional workflow, but if you have multiple samples, I think it's definitely a small price to pay, because doing the Gexscope workflow 6 times, it's not easy. This time you can just do 1 and have your 6 samples. So you save time and money. Okay. Now I'm going to present you some of the data that show the performance of CLindex labeling. So first of all, the efficiency of labeling, the effect of labeling on gene expression and cell clustering, which is very important. We're also going to go into the equivalence in labeling between different cell type. Like when you have a population of cell, you don't want to have a preferential labeling. And between different species. We're also going to talk a little bit about cell viability. And finally, we're going to look at the performance of CLindex against an antibody-based technology, also known as cell hashing, which is basically the gold standard those days in multiplexing. Okay, so the first experiment. So we use 4 different cell lines, so 4 hematopoietic human cell lines. And they were all labeled with different CLindex tag. All the cells were then equivalently pooled, and we ran 1 Gexscope workflow. And you can see on the pie chart at the bottom left of your screen that the cells were well represented. So they basically have a quarter of the read each. You have some of the cells that were multiplex, so that means that 2 or more cells were in 1 well. But now you can tell because they are tagged. And you can see that some of the reads are undetermined, so you cannot really say which tag was there. It's on 1% of them. But basically, the labeling efficiency reached above 97% in this particular experiment, which is very good. On the right side, you can see the distribution on a t-SNE plot of the 5,000 different cells that were captured. And you can see that the 4 different cell lines basically cluster by themselves. We then look at the effect of labeling on gene expression and clustering. So it's basically the same experiment, but we added a no labeling control. So we use the same 4 cell lines, all hematopoietic cell line from human. And instead of labeling them all before pooling, we basically just pool them. You can look at the different metrics at the bottom here on the table in blue. The same, very similar metrics here. So you look at the number of cells that were captured. So it's pretty much around 5,000 in both cases. How many reads went to each cell. So again, very similar. And you can look at the bottom at the total number of genes. So 25,000 genes were identified in both experiments. So seem pretty equivalent. You can then look at the transcription of the bulk. So if you look at those 25,000 genes that were identified and the level of expression that they show on the 4 cell lines, so you can see that between the unlabeled and the labeled cell, it's pretty much the same. The Pearson correlation was above 99%, which is excellent. On the right side of the slide, you can see that the control cluster again in 4 different quadrants on the UMAP plot. And it's the same for the CLindex label. And on the third graph at the bottom there, you can see that they overlap almost perfectly. Also, a fun thing that you can do with the CLindex tag is you can basically color your sample by the tag. So you can basically find back which cell line belongs to what. This is the last graph. In this experiment, they really pushed the system. So they used the 16 different CLindex tags in one experiment. So again, they used the human CCRF cell line, and they divided in 8 different pool of cell, and they all receive an independent CLindex tag. They also used a mouse testis cell that they did the same. They basically divided in 8 different pool and labeled them, and they all pull everything together, 16 different samples. They loaded the high-density chips for this particular application, since we have a lot of cell, a lot of different indices. This chip has 250,000 microRLs and can capture, in this case, for example, 45,000 cells, which is pretty significant. You can see all the cell clusters. So in green, we have the human cell. In blue, we have the mouse, and in yellow, you have the multiplex. So when you have more than 1 cell per well. There's about 11% of them in this experiment. And you can see that in the pie chart at the right, bottom right of your screen. This is also the distribution of every sample. So basically how many cell we have captured. And it's vary from 4 to 7%. So again, it's pretty equivalent. There's no bias. Same experiment. So it's just to show you on another graph the distribution of the cell. You can see the number of cells that were captured for every sample. So an average of 2,500 per sample. You can also see that the clustering is not affected. It's the same for the different cell type. And for sure, the human and the mouse cluster differently. And we also assess the cell viability. So before labeling, you can see that the human CCRF viability was about 97%. And after labeling, we recorded again the viability, and it it goes from 92 to 97%. So some of the samples were affected, but most of them were not. And lastly, I want to show you the performance of CLindex compared to the gold standard, which is an antibody-based technology. So for this experiment, we use mouse bone marrow. So we divided the sample in 3 different categories. So the no label, so no processing, CLindex labeling, or antibody-based labeling. We went into the JEXscope workflow again for single-cell RNA-seq, and we look at the clustering of the cell. And this is the graph at the bottom of your screen. So in gray, you have the no labeling. In pink, or salmon, you have the CLindex labeling. And in purple, you have the cell that were labeled with the antibody. And what you can observe in this category is that some of the cell in this population are disappearing or reduced. So the mature and the pro-B cells are slightly reduced, but you can see that the erythroblasts are greatly affected. You can see again in the plot that look at the transcription between the 2 bulk experiments. So the no labeling versus the CLindex labeling, the Pearson correlation is above 99%. So that means that the transcripts are pretty much the same between the 2 experiments. But the Pearson correlation between the no labeling and the antibody labeling really took a drop here with 44%. And you can think that it's because of some of the cells are not represented, and the transcription level is affected by that. Okay, so let's resume the different advantages of using multiplexing in CLindex for your RNA-seq experiment. So CLindex is unbiased. There's no impact on the gene expression or the cell clustering. It's a great cost reduction, up to 60%, depending on how many samples you want to multiplex for your experiment. You can multiplex up to 16. It's pretty fast. You can do it under 30 minutes. And it's a great time saver for downstream because you only have to run the workflow one time for all your sample. It's a good application for our high-density chip. Again, the high-density chip has 250,000 wells, and you can load over, or you can capture over 30,000 cells. It's universal, so the chemical reaction is compatible with different cell type. It's not going to reduce some cell in your population or advantage some of them. And it's species independent, so you can use the same kit for if you have in your lab a mouse model or a human model, you can use the same kit. You don't need to buy two kits. And it barely affects the cell viability. And of course, the kit contains all the reagent that you need to perform the experiment, so you don't need to go chase reagent from a third party. And on that note, I would like to thank Dr. Li. He's a professor of chemical engineering at Nanjing University. He helped Singleron developing this product. So thank you. And thank you all for your attention.
Thank you very much, Julie. That was really nice presentation, clear and cut. So we're back to the Q&A sections. We have also some questions to Julie regarding the CLindex methods. So one of the questions is, does the CLindex work for formalin or methanol fixed cells?
Oh, that's a good question. I think we did not test this. Something definitely to add to the queue. But yeah, I would not see why it would not work.
And another related question, have you tried to label other species, such as, so others other than human and mouse?
Not at the moment, but if you are interested on suggesting some model system that you would like us to test, we're open for that, for sure.
Mm-hmm. And also, I think there is also a question regarding the CLindex. So the question is, CLindex-based labeling, is it mainly relevant only when versus antibody labeling? So what are the differences? And yeah, I guess that is the main question.
Well, there's many differences between the chemical quenching that is used in CLindex and the antibody-based technology. They're both basically doing the same thing at the end of the day, but one is more efficient and less biased than the other. So you can imagine for the antibody-based technology, you will need a set of antibody that is species-specific. And because you target only some type of protein at the surface of the cell, you may think that you will see more bias inside a population of cells. With CLindex, it's a group at the surface of the cell. It's not a special protein that is recognized. And I will add to that this group is basically at the surface of every cell. So it's why we think that the product is species agnostic. I also add that the link or the bound that is between the CLindex oligo and the cell is covalent. So it's really a tight attachment. And since it's a chemical reaction, it happens like very fast. With an antibody-based technology, you will have to incubate your cell and the antibody for a longer period of time.
Okay, excellent. Also, another question is, have you evaluated the CLindex labeling on cells from primary tissues? Is it also compatible with nuclei multiplexing?
Since the nuclei also have a protein at its surface, it should be compatible, but this is something that we are still working on.
And the other part of the question is, have you evaluated working directly from tissue, so to label from cells from the tissue?
We did not, but this is not something I would suggest to do. because you really want this the oligo to have access to all the cell. And I can imagine that if they are still all bind together in a tissue, that it may limit your access to some of the cell.
But do you see that this could work well after this single cell suspension? So if you would be able to get a very clear single cell suspension after the dissociation, then this step can be added, correct?
Oh, yeah. So after the tissue dissociation, yes, CLindex is compatible.
Okay, great. And due to the limitation of time, we might have just one last question. One last question regarding the because you introduced also the high density chip. So the question is, what is the expected multiplet rate with the high density chip? And is increasing cell load increasing the chance of multiplets?
Yes, so of course, more cell you load on the chip, more chance you have to have two cells in the same well. And more multiplexing you do, more you can identify those wells. So you can remove them from your analysis. Of course, you're losing sequencing data, so you want to act here with some moderation on the loading of the cell. You don't want to lose all the data. So yeah, it's an equilibrium, I would say, that you have to find. We can offer some recommendation, of course.
Okay, yeah, so due to the limitation of time, we will end today's webinar here, and we have listed our contact address and our official website, and we welcome to hear from you soon.